if these negative thoughts and circuits underlying these are active while ketamine is on board, then those circuits, hyperactive circuits, will be weakened, reducing these negative thoughts and thereby potentially reducing depression.
3, 2, 1, liftoff.
Welcome back to the Ketamine Startup Podcast. I'm Kim Ko, and this is where we talk about building a ketamine therapy practice, industry insights, and what's shaping the future of psychedelic medicine. My co-host Sam Ko sits down with a basic scientist whose research may have you rethinking some of what we take for granted in the outpatient ketamine therapy space. I'm not going to give away too much before we jump in, but if you've ever wondered why some patients don't seem to respond as well, or why results may plateau over time with ketamine therapy, this conversation offers a perspective worth sitting on. Before we start, though, a quick reminder. The information shared in this episode is for educational purposes only and does not constitute medical advice. Treatment approaches discussed should be evaluated with your specific practice context and regulatory environment. Now here's Sam.
Welcome back to the Ketamine Startup Podcast. I'm Sam Ko and your host for today's episode. Normally, we spend a lot of time on the show talking about the clinical and business side about ketamine. But today we're going to go upstream and we're going to get into the basic science. My guest today is Dr. Roberto Malinow. He is an emeritus professor at UC San Diego, a member of both the National Academy of Sciences and the National Academy of Medicine. He's one of the world's leading researchers on synaptic plasticity, NMDA receptor biology, and his work has been cited more than 30,000 times. He published a perspective piece recently titled How Ketamine Works, an Actionable Hypothesis. And when I came across this article, he was kind enough to send me the PDF version of it for private use. And I'm really grateful because it has a very different perspective than what we may normally be used to as far as in the outpatient ketamine clinic setting. So I wanted to bring him on the show. And so with that, Dr. Malinow, welcome to the show.
Thanks for having me.
My pleasure. I am curious because I was reviewing your CV and I want to rewind way, way, way back. And so I noticed the first thing that stood out to me was you went to Reed College, and that is in Oregon. And I also recall that's also where Steve Jobs went to school for a couple of years. So I'm really curious, like, how did you choose Reed College? Because for those of you who don't know, it's a very different type of education than your normal standard educational college experience. So tell us a little bit about that.
Well, I'm a son of immigrants, and we lived in Portland. And as immigrants, my parents wanted to keep me close, and so Reed College was a place that I'd seen, and I'd had some friends go there, and so I had a very positive view of it, and so that's where I ended up going.
And when you were in college, did you know you wanted to get into, the synapses and neurons, or how did this come about? Because you've
Not
become
at
an
all.
expert in this.
Yeah, well, that was somewhat later, but I studied math in college, and I don't think I was particularly good at it, but it was very challenging, and I really liked it. So it just helped with problem solving, I think, in general. But that's what I studied there, and yeah, synapses came quite a bit later, but I studied math there.
Okay, great. And it looks like you went to NYU School of Medicine and got your medical degree. And then you also went to UC Berkeley and obtained a PhD. So this is really fascinating because you have both a medical degree and a PhD. How did that come about?
Well, I was accepted at NYU. It was one of 20 medical schools that accepted me, but one of 20 that the one of the 20 that accepted me. Um, and I started in the MD PhD program there, and but I finished out the MD in NYU and then ended up going to Berkeley for my PhD.
And when we spoke earlier, you mentioned that you don't really do the clinical aspect of medicine. You're focused more on the basic science, although you did do a 1 year internship as in internal medicine. Did you know that when you first started down this MD PhD that the basic science research was going to be your route, or were you planning on
Um,
doing clinical medicine?
No, I think that I always wanted to do research. I had done research every summer during my college time, and I really liked it. And so I knew that I wanted to do research, but I didn't really know which field. And so I kind of went to medical school to find out all the different fields that were available, and so that's what happened.
Yeah, and tell us the story. How did you get into Synapses? You mentioned it came later in your career. So how did
Yeah.
that
It
interest
was
begin?
actually during one lecture in medical school, second year, where my professor Rodolfo Llinás was talking about synapses, and he had this way of being very excited about synapses, and so it was very exciting to hear all about the things that synapses could do and how complicated they were and what a puzzle they were really. And so that's how I initially started my path down the study of synapses.
Yeah, and what's fascinating is with ketamine, and we're going to be talking about that. So, tell us your understanding of how ketamine impacts the NMDA receptor, because I know you have a little bit of a different perspective with your expertise.
Well, I don't know if it's that different. Ketamine blocks channels that are open, NMDA channels that are open, and so they have to be active in order for ketamine to really have an effect. And so that's one of the basic ideas that how ketamine works. And there are a number of drugs that work like it. There are many drugs that act on NMDA receptors, and NMDA receptors have a lot of different ways of getting blocked. And ketamine and drugs like it are have a specific way of blocking it.
And tell me about um, the disappointment center, because we had spoken about this previously on our telephone conversation before the interview,
Right.
and you mentioned the disappointment center in the brain. And I'd love to hear you expound upon that.
Sure. There's this idea that has been around for a few years now that this little part of the brain next to the thalamus called the lateral habenula acts like the disappointment center. And this part of the brain inhibits dopamine centers. And so it's thought to be that way, how it makes you feel bad, why you feel bad when you're disappointed. And it's important evolutionarily to have a disappointment center because it's part of reinforcement learning, really. It's learned by your mistakes. So if you make a mistake in making some choice, you want to feel bad about it. And so if you don't get what you expect, you get disappointment, you feel bad about it. And the next time you're given that choice, you'll choose something else. And so it's important in evolution to have that. But it's thought that potentially thought that depressed people might have this disappointment center be hyperactive, that it's too active or too easily activated, and that could potentially be a significant contributor to depression.
And your paper was fascinating in that normally in the clinical outpatient setting, a lot of clinics, we focus on this concept of set and setting, where we want the patient to have a, kind of a positive mindset or not have any negative emotions during and immediately before the ketamine infusion. And then we also optimize the setting with like eye shades and really pleasant music and maybe some pleasant odors like lavender or whatever it may be. But the thing that really stood out about your article was we may be facing or doing it in a different way, or we may be able to leverage the set and setting in kind of an opposite of way of what's being done now. So I'd love to hear your thoughts on that.
Sure. Let me just bring out the basic elements of this hypothesis, and I think it'll be clear why set and setting might be relevant. So the hypothesis has three basic elements. The first is that in order for ketamine to produce a lasting change, it has to do this through neuroplasticity, through some kind of modification of brain circuits. And how can ketamine do this? Well, we found some 10 years ago that drugs like ketamine could weaken hyperactive circuits, hyperactive synapses, and synapses that are inactive wouldn't be affected by ketamine. And so, how are hyperactive circuits relevant to depression? Well, some believe that a significant contribution to depression are negative thoughts, thoughts that of guilt, of worthlessness, ruminations, things that are difficult for depressed people to turn off. And so, if these negative thoughts and circuits underlying these are active while ketamine is on board with a person, then those circuits, hyperactive circuits, will be weakened, reducing these negative thoughts and thereby potentially reducing depression. So, those are the basic three elements: neuroplasticity, weakening of hyperactive circuits, and specifically with respect to depression, it would be weakening hyperactive circuits responsible for negative thoughts.
Yeah, and you even mentioned in the paper that there was a study for patients with PTSD who were doing something called written exposure therapy, and how in that research where they were priming the patients to write about these traumatic experiences. Um, and what was interesting was that for that paper, they were not doing it during the infusion, but in between the sessions. But what I'm
Right,
hearing
right.
you say is actually during the ketamine infusion. So, the standard protocol is zero point 0.5 milligrams per kilogram up to maybe 1.5 mg per kg in a 40-minute infusion, that it may be beneficial if we activate these synapses and these connections where the patient is thinking about these feelings of guilt or hopelessness or the trauma or whatever it may be that's ailing them.
Right, right. Yes, and as you know, when someone's under the influence of ketamine, they get kind of high, so it's kind of difficult to ask them to do things. But I believe that this written exposure therapy might have just brought up those ideas in their mind and kind of got them going, so that when they did take ketamine, those circuits were more likely to be activated. Um, so I think that may be how it was working, and maybe including written exposure therapy in depression rather than PTSD might be something worth looking into. Although I think the best way to test my hypothesis won't necessarily be in depression research, but maybe in treatment of chronic pain, which I know you do that some. But we can go into that if you want.
Yeah. So the idea with the chronic pain would be stimulating whatever causes that pain. So if it's, I don't know, for example, peripheral neuropathy, then doing something during the infusion where
Exactly.
uh, that stimulus is created, because then it'll activate those neurons or synapses to
Right, right.
allow the ketamine to target, is what I'm hearing.
Exactly. Exactly. Yes. And in theory, I mean, it would probably only work with certain kinds of pain, but if you can access that pain, I think it'd be easier to do that than, say, accessing these negative thoughts. But yeah, I think chronic pain is a great place to start, and I hope the clinicians out there will try it out. It's relatively easy to do. I would think that you isolate what the source of the pain is, and as long as that physiological effect, like inflammation and whatnot, has already subsided, if it's thought to be a sensitization of central pathways of in the spinal cord or in the brain, then those pathways are what you'd want to activate while a person's on ketamine, and thereby weaken those pathways and potentially reduce the chronic pain.
Hmm. And, it reminds me because I actually had a patient who I was treating at our clinic, and she suffered from panic disorder and anxiety attacks, as well as depression. But in this scenario, the patient had her car broken into like immediately before the treatment, and she was very stressed out. She's like, they stole this and they stole my purse and et cetera. And she was in this really highly agitated state. And I asked her, I said, do you still want to proceed? Because it seems like you're, kind of activated and it may not be the optimal setting. And she said, you know what, I still want to do it. And the experience, and this is just anecdotally, I remember she had a really bad trip where she was scared during the infusion, and those neurons or those patterns were activated. And I think we had to stop the infusion maybe midway, 20 minutes in out of a 40-minute session. And for me, when I saw the patient have such an adverse reaction, in my opinion, to that, anxious state, it seemed to like make her symptoms, at least in the short term, worse. So I'd love to hear your thoughts on that.
Hmm. Well, the brain is complicated, and it's hard to know exactly how all these things pan out. I was hoping you'd say, "And she was miracle, she was fixed," but yeah, I don't know if there've been studies to see how patients, depending on how their actual experiences during the uh, the ketamine, how that has a predictive effect on the outcomes. I'll say one thing that is a little bit relevant, which is you may know this literature that depressed surgical patients were giving ketamine while they were unconscious, and ketamine did not improve their depression. And so, presumably, the brain has to be active in order for ketamine to have an effect. And who knows what kind of activity would be necessary, but at least that's consistent with the idea that you will modify circuits that are active, and they need to be active in order for ketamine to really have an effect.
Yeah, thank you. I remember coming across that study. I believe it was published by Dr. Boris Heifets out of Stanford, and they put patients under general anesthesia and then compared, one group of ketamine receiving patients and another group who didn't, and they found that the depression symptoms, the scores remained similar. And I do recall that both groups actually had drops in depression,
Hmm.
right? The non-ketamine group and the ketamine group. So that was very interesting. Which some people say, well, maybe there's another component, like you said, like maybe the brain has to be active for certain areas. What would if you could design a study on humans, and just for clarification, Dr. Malinow focuses mainly on the research, basic science side. So if there were, let's say, a clinician researcher who's actively, working in this, what could a study design look like?
Sure, sure. Well, as I mentioned, I think the easiest area would be in chronic pain, because there you can activate the circuits more easily than other areas. But I think um, in depressed people, maybe you could get them to write about their depression and the triggers and what makes them feel bad, and have that be sort of in mind, at least when they go into the ketamine treatment. So that's whatever makes people have these painful stimuli or painful thoughts active while they're on ketamine might be a way to test this. But as I say, I think the easiest would be in chronic pain.
Hmm. Yeah, that would definitely be easier because you are right. When someone is under the effect, the person's ability to focus on certain thoughts or memories will be quite challenging versus the, biological stimulus of creating pain that might be much easier to actually stimulate versus having the patient consciously try to control
Right.
where they're going.
Yeah, and some basic science experiments have been done with chronic pain in this respect. And drugs like ketamine, not ketamine, but a drug like ketamine was used to reduce pain, but it was reduced only if the drug was given while the painful stimulus was delivered. And so the experimental protocol was to induce some kind of chronic pain and then um, activate that pain while this drug is on board. And only in those animals where the pain was being activated when the drug was on board did the pain resolve or was it reduced. And so I think there's some evidence to support this kind of idea.
Yeah. And I want to zoom out for a moment because I have all of your knowledge and wisdom and experience. So there was a quote, and I don't recall who said it, but they said something like to to study neurons to understand consciousness is like studying words to understand Shakespeare. And so I'm curious to hear, you've studied neurons, you've studied synapses, and what are your thoughts on consciousness and how the synapses, if you just have, this may just be pure speculation or
Yeah, well,
hypothesizing without, any hard proof, but what are your thoughts on the connection between synapses and consciousness?
Well, they need to be active in order to be conscious. I think everyone can agree with that. But I think the connection between synapses and neurons and the higher abilities like consciousness is still pretty far away. And as you know, neuroscience has a way of really focusing on different levels, either the molecular, the cellular, the circuit, the behavioral, and all the way up to consciousness. And over the years, it's been very difficult to move from one level to another, which is really what you're asking me. You're asking me to move from synapses and circuits all the way to consciousness. But nowadays, there are tools that are being developed that could potentially, I'm not sure how soon it'll be to consciousness, but it certainly is able to ask questions about which circuits are underlying which behaviors. So, this is a technique you may have heard of called optogenetics, where you can deliver specific proteins into neurons that make them sensitive to light. And so, if you shine light, it'll activate those neurons and those pathways. And it can do this while the animal is behaving. And so, you can see what effect activating a certain set of neurons has on behavior. So, at least that's starting to make that leap between one level and another. And it's really quite an achievement because, as you know, the brain is like the big muddle of nerve cells. Every part communicates with every part, and it's very difficult to isolate specific pathways and specific groups of neurons to really see what effect they have. But with this new technique that's now been around for over 10 years, I'm sure they'll get the Nobel Prize on this. And because it's able to really ask questions about activating and also inactivating certain pathways and what effect it has on behavior. So I don't think I can answer your question about the role between neurons and consciousness, but I can tell you that neuroscientists are thinking about this, and it'll come someday.
Yeah, and I want to double click on optogenetics and how certain light patterns, because this is, I'm not really familiar with it. So, what I'm hearing you say is that by flashing certain patterns of light into the retina can stimulate certain emotions.
No,
Is that what I'm
not
hearing? Or
no, not exactly. So this method is one whereby you use a virus to deliver DNA, which encodes a protein, into neurons. So now these neurons are expressing this protein. This protein is light sensitive. And so when neurons express this protein, then they become light sensitive. So they're not neurons in the retina, they're just any old neuron in the brain. And so if you shine light, there's certain proteins that will activate those neurons. And so you can selectively activate those groups of neurons that are expressing this specific protein. And so in that way, you get specificity by delivering this light sensitive protein to a certain subset of neurons. And you can target different subsets genetically and a bunch of other tricks. But that's basically optogenetics, where you sensitize nerve cells to be light sensitive. And that permits one to do a lot of really interesting experiments.
Hey, listeners, this is Kim, and I just want to acknowledge that you're here for a reason. Maybe you're stuck in a cycle of long shifts, feeling a little burnt out, and wondering if there's any other way to practice medicine, especially on your own terms. We've been there, and that's exactly why we created this free checklist, your step-by-step roadmap to starting a ketamine clinic without those costly mistakes. Grab your copy now at www.ketaminestartup.com/checklist. It's completely free because we want you to succeed, not struggle. And hey, if you're driving or mid-workout, don't sweat it. Just check out the show notes for the link when you're ready. And again, that's www.ketaminestartup.com/checklist. Now let's get back to the episode.
great. Thank you for that explanation. And I want to go back to depression. And from your perspective, what's the most important thing that we don't yet understand about depression from your perspective?
Well, that's quite a large question, but I mean, I still think that the circuits that underlie the the feelings and thoughts that are responsible for depression are not well understood. And one can have models of depression in animals and look to see. And in some of those models, in fact, they've found that this part of the brain that we talked about,
Lateral
uh,
habenula?
the lateral habenula, the disappointment center, that was hyperactive in models of depression, consistent with the idea that uh, that part of the brain could be significantly causing depression. And in fact, in some experiments that we did, we inactivated that part of the brain and were able to reduce um, depressive symptoms. And so, what we don't know is what parts of the brain are hyperactive, what parts of the brain are not active, and really how we can target those. Lots we don't know.
You mentioned something at the end of the paper that you sent over my way. And tell us about the dystopian scenario that you that was to me when I read that portion, I was like, "Oh, it's quite a shift." So I want to hear more about that from you.
Yeah, this is a pitch that I have for anybody who wants to make a dystopian movie, which is that the idea would be that if you can give a drug like ketamine, one that doesn't make people high because they'd be able to tell. But if you give people a drug like that and then expose them to something that you want to wipe out from their mind, and I brought up the idea of liberal democracy, which is in the news these days. And if one wants to wipe that out from a population, potentially you could do that. And I mean, in theory, you could wipe out any idea, and it would be kind of a mix of the eternal sunshine of the spotless mind with some new age kind of science fiction. But yeah, it's anybody want to make a movie? I'll let you know how it could work.
Yeah, I remember reading that. I was like, okay. I mean, that would be a pretty scary movie where large political governmental organizations could wipe out memories from, a whole group of citizens. Um,
Right.
and yeah, that mean that's wild to think about, but also very possible. Maybe not the drug ketamine or potentially
Right.
ketamine, but yeah, it was very fascinating.
Yeah, it would have to be something that people didn't obviously know that they were being affected by a drug, but um, yeah, who knows? Maybe even it could be. But uh, yeah, it's these crazy ideas you have when you're falling asleep.
I love it. What's something that you've seen in your interactions with clinicians, and you having the PhD background and the research background? What's a common misconception that clinicians may have about synapses and neurons that you frequently are like, oh yeah, for some reason that's just something that they don't understand, and maybe not everyone, but yeah, have you seen any misconceptions come up?
Um, well, I I don't think clinicians really think too much about synapses. Um, because it's not directly relevant to how they practice medicine. Um, so I don't really have much to say about misconceptions. Maybe if I think about it a little bit more, I could, but I think yeah, I think they have other things to to think
You
about that are more relevant.
what's one thing that surprised you as you began your trajectory down this pathway that surprised you personally?
Surprised me about
About studying synapses from back in medical
right,
school
right,
when
right. Well, what's amazing is how plastic they are. There's so many forms of plasticity in synapses. There are these little engines that can really be modified, and it's probably the basis for all our thoughts and feelings are how these synapses work. But as you mentioned, the connection between synapses and consciousness or behavior is still pretty far away. But yeah, someday there'll be more and more connections between synapses. And I should mention that even though clinicians don't think about synapses, uh, that's where most of the neuroactive drugs work is at synapses. And so certainly the drugs that they use act on synapses, but exactly how those get modified by the drugs is not is still not very clear.
yeah, you're absolutely right. I mean, we're prescribing SSRIs and, dopamine agonists and benzodiazepines, and those are all working at the synaptic level. So I think it is important for clinicians to recognize neuron synapses, although we may not directly explain it or think about it, but with the things we are doing have a direct impact on the neurology and the synaptic level.
Yeah, I think that probably every psychiatric drug acts on synapses, and we still don't know very much how those work. There, you give this drug that works on the whole brain and doesn't really target any area in
Mhm.
particular, but it has these magical effects. So how that happens, we don't know. Um, so one thing to mention is that ketamine really has a rapid effect, as you know, that its effect on depression can start almost immediately. And so, whereas other standard treatments take 2 to 3 weeks for them to work. And so ketamine in that way has been revolutionary in the field of psychiatry because it can reduce suicidality acutely. And so people don't have to be, in the psychiatric wards for 2 to 3 weeks for the antidepressants to work. But it really challenges us to ask, so how can ketamine work so quickly? And what I'm proposing is that it's working by selectively weakening some circuits, and we can see that in uh, in the lab in brain tissue, where we can ask specifically, what does this drug do to synapses and synaptic transmission? Um, and it does this very quickly. It can do this very rapidly, and so that's part of the basis for thinking about how ketamine works. That you have to really think, how can it work rapidly? So that's the idea, would be that it would work rapidly by rapidly weakening hyperactive circuits.
Absolutely. And, you bring up a really good point about how rapid it can work. And then as you were speaking, it makes me think about how certain experiences in our life can work rapidly and create neuroplasticities. For example, if someone has, I don't know, a car accident, let's say, like that is going to rapidly change the brain. Or if someone has, I don't know, a family member or a friend that passes away suddenly, or it's just some major event, winning the lottery, something like that. That
Right,
how
right.
these
Yeah.
one-time events can, in a way, rapidly change the brain as well.
Yeah, this is a party trick that I used to have, which is, okay, Sam, I'm going to change your brain. Okay. I'm going to change your brain. Right now, your brain doesn't know certain things, and I'm going to change it. Something I can ask you, something that you may not know. Where was I born? And you don't know this, and your brain doesn't know this, but I'm going to modify your brain so that when I ask you next time, you'll know. I was born in Argentina. Boom. Your, your brain's been changed. Now I ask you, where was I born? And boom, you know. So that's my little party trick.
What I love about that is, it's learning at its essential core, right? We don't know something, and then we know something. Now the memory has changed, i.e., there's been neuroplasticity. So, someone could argue there's been remodeling of the brain. So I love that party trick, and I do want to let you know, I actually know or knew where you were born in Argentina
Okay.
because I did my background research.
Right, right. Okay.
Um,
Well, then maybe I didn't quite change it, but I'm sure I could find something you don't know.
Yeah, it's really interesting to think about how the brain changes as we learn. And maybe these therapies like ketamine is working in that same pathway. Like you said, something, these are these hyperactive portions of the brain
Yeah.
that are being altered in a rapid fashion. And then, why isn't that happening with the SSRI or, the other ones that can take weeks to months to kick in? Is really fascinating to think about.
Yeah, and as you mentioned, neuroplasticity, one of the key elements of that is the NMDA receptor. And the NMDA receptor plays key roles in enhancing transmission through plasticity and also weakening transmission. And so NMDA receptors have key roles to play, and how they get activated, how they get blocked, um, are really important elements to learning and memory and to neuroplasticity. I mean, we and others have shown that you can in a sense form memories in animals, and then selectively weaken those memories, and the animal completely forgets. And we did this in a paper a number of years ago using optogenetics, where we delivered this light sensitive protein to pathways that went into the amygdala, which, as you know, is important in forming fear memories. And what we found was that if we paired a light stimulus to this pathway to activate these neurons going into the amygdala, if we paired that with a foot shock, then subsequently, when you gave that stimulus, the animal would freeze. And so the animal essentially remembered the foot shock just by that pathway having been modified. And we could go further, and we could weaken that pathway by activating those neurons in a specific way, which weakened synapses, which we know weaken synapses. And in that way, it essentially removed that memory, so that when you activated that pathway again, the animal kept on moving around and couldn't remember it. And we could reactivate it to make it strong again, so that then the animal remembered again that foot shock. And so we know that the NMDA receptor is critical in forming those kinds of memories. So we do know quite a bit about how memories are formed, especially those memories that are dependent on NMDA receptors. And we were able to turn on and turn off memories, really leveraging what we know about synapses and what we know about NMDA receptors, and something about what we know about the brain pathways. So this was a paper that had quite a bit of interest in it.
I also want to expand upon that and get your thoughts on the glutamate neurotransmitter, because I remember reading somewhere that, and please correct me if I'm wrong, that glutamate made up 90% of the neurotransmitter in between, in the synapses. So I'm
Yeah,
curious, like, we not only have the NMDA receptor, but then we have to consider the glutamate neurotransmitter itself.
right. Yeah, glutamate is thought to mediate, I think, 90% of synaptic transmission in the brain. And there are a number of different glutamate receptors. And we know pretty well how a number of these receptors work. But the problem is that the whole brain uses glutamate, but it uses that in specific pathways. And so it, the brain can activate specific parts uh, by activating certain pathways. Um, and it does that primarily through glutamate. And so it uses this key neurotransmitter everywhere, but specifically how you get certain behaviors is by specifically activating some pathways and not others. And by the way, the other 10 or so percent um, uses GABA, which is an inhibitory neurotransmitter. So glutamate is an excitatory transmitter, and GABA is an inhibitory transmitter. And so those are the main ways that pathways are activated or inhibited by neurotransmitters.
Yeah, I remember learning in medical school, I think it was like our embryology class, and they were talking about how the central nervous system was formed, I believe from the neural crest tissue. But then I remember learning that the enteric nervous system also comes from that same origin of neural crest. So a lot of these neurotransmitters we're talking about, whether it's serotonin, dopamine, glutamate, it's not only in the central nervous system, it's also in the gut. And so
Yes,
with SSRIs, for
absolutely.
example, it's going to, some people will get nausea or diarrhea or other GI symptoms. Um,
Right.
so are there synapses, and, you may or may not know this, but are there synapses as well in the enteric nervous system, to your
Yes.
knowledge? Okay.
Yes. Yeah. Yeah, there are circuits that are completely contained within the gut, so they don't even directly communicate with the spinal cord or the brain. They control local things, and they do that by using the same neurotransmitters that are used in the brain. And so, yes, indeed, serotonin is one of them, and in fact, it's thought that serotonin used there, and there might be gut microbes that affect serotonin or even make serotonin, and that can affect both the gut and the brain. So there is this big idea nowadays that there is an important connection between the gut and the brain, and that's going to be up to future neuroscientists to figure out exactly how that works.
Yeah, it's fascinating. And, there's that saying like trust your gut instinct. And so maybe we are relying on our enteric nervous system as part of the whole decision making process as far as, at the synaptic level.
Right, right. Yeah. Yeah. The brain is complicated enough, but now if you bring the gut into it, I don't know how that uh, is going to simplify things, but yeah, that's what we have. We're a very complicated machine, and all the parts have different ways of working on their own and working together.
Um, Given your background in, just being so involved in the scientific literature, what's one or two papers that you can think of that anyone who's working in the ketamine space should ideally read? Is there anything that comes top of mind, like, oh yeah, this paper by XYZ author? Is there anyone that's coming up for you?
Um, I can't think of any specific one. I mean, there are review articles about how ketamine works. Um, I mean, ketamine's been around for many years, as you know. And so there have been a lot of studies really on on ketamine. Not so much how it works in depression, although now that's been used for 15, maybe 20 years probably by now. So there is quite a literature on that. Um, but yeah, I'd have to think about if there's any one specific. I don't know if there's like a Scientific American article on ketamine there. There probably should be, but because quite a bit is known about it. But anyway, yeah, I'm interested to hear what people have to say about this specific hypothesis that I have about how ketamine might be working.
Yeah, and I hope all of the listeners and viewers will definitely read through this. We'll put a link in the show notes so that you can easily find it. Um, I would love to hear your thoughts, because there's this whole psychedelic renaissance that's happening. That people are studying ketamine, but then they're also studying MDMA or psilocybin or DMT or whatever it may be. And there's some interesting research actually happening at UC San Diego, I believe at the Center for Psychedelic Research, where they're studying these various compounds that are Schedule One and seeing how it can affect pain. So, what are your thoughts on these other upcoming, not FDA approved medicines,
Right.
substances that are coming out?
Yeah, there's some interesting basic science with these drugs that is being done these days. Um, I know that one lab has found that the actions of psilocybin might be through the disappointment center,
Mm.
actually, that it can change the activity patterns that are occurring in the disappointment center and effectively quieting that part of the brain. And so I think it'll be interesting to see how that pans out. and whether others are able to replicate those findings. Because as you know, in science, it's important that we make hypotheses, test them, and then have other people test them. Um, and that's how really progress is made in the field. But I think looking at how these psychedelic drugs affect different parts of the brain, and specifically the disappointment center, I think will be quite an interesting set of studies for the future.
Yeah, absolutely. Definitely more research is needed. And it also makes me realize that these other drugs that are being studied, they also create rapid changes where some of these
Right,
studies are
right.
showing, they'll do one dose of psilocybin and then they'll do a 24-hour, 48-hour, 1 week check-in. And the patients, the research participants have dramatic drops. So I wonder if the ketamine hypothesis about stimulating certain, like if someone is undergoing a psilocybin research, I wonder how stimulating chronic pain during that psilocybin session or MDMA session, whatever the researcher is studying, could also play a role. Like maybe those areas need to be stimulated for psilocybin, et cetera, to be effective as well.
Right.
What do you think?
I don't really know how psychedelic drugs would work, but I think you're proposing that negative thoughts would be preferentially weakened by psychedelic drugs, and that's certainly possible. I mean, they have direct action on a different set of receptors than ketamine, but the mechanisms could be similar.
Yeah, there was a really interesting researcher clinician in Mexico. This is like, many, many years ago. I believe his name was Dr. Roquette, Roquet, from Mexico. And he would bring in patients and have them take LSD, ketamine, etc., psilocybin, and then he would expose them to really loud music, really unpleasant experiences, visual images that were kind of horrific. I don't know if you've ever heard of this researcher,
No, no,
but
I haven't.
when I was reading your paper, it made me think about that clinician, like how he was doing it way back in the days. And there was controversy associated with him, but he did take in a similar approach where he would expose them intentionally to these really negative types of stimuli. And
Right,
it was kind of like a shock therapy in a way where it was
right.
very intense for individuals.
Right. Well, that's fascinating. Yeah, I'd be interested to see what the results of all those were. But it's certainly a different take on set and setting than is currently being used in a lot of this train in a lot of these ways these days.
Yeah. The sun is setting on our conversation, and I want to start landing the plane. So I want to ask you some fun, rapid fire questions. And the first one I have for you is if you could go back in time for a day, where and when would you go and why?
Hmm. Maybe I'd go back to when the first neurotransmitter was discovered by I don't know if you know this German fellow Otto Loewi, and the first neurotransmitter was from the vagus nerve, where they squirted out the stuff they called Vagusstoff, the Vegas stuff, uh, onto the heart, and it reduced the heart rate, and so they found that that was the first neurotransmitter. I think it would be very exciting to be in that uh, laboratory where they initially showed that boom, you do something and something changes like that, and so that would be very exciting to go into those times when you make real discoveries. That's the most exciting thing for a scientist is really making discoveries.
Do you have a hidden talent or hobby that most people would be surprised to learn about you?
I play a lot of tennis. So some people know that, but maybe that changed your brain right there because you didn't know that. We have an example. Um, I I play guitar sometimes, and yeah, playing tennis. That's my hidden talent, I guess.
All right, very good. And let's say that you were to, I don't know, go back and rewind, and for some reason you could not get a medical degree, you could not do PhD level research, and you were to just, let's say, have a completely different career. What would you do instead?
Mm, I think making movies. I have those ideas. I've always had some idea of making movies, although I'm sure it's a lot different than my view of making movies. But I've always thought that being able to create some different reality would be lots of fun.
Yeah, and maybe that dystopian movie that you're talking about would be
Right,
like,
right, yeah,
a good
yeah.
one.
Any Hollywood people out there, give me a call.
Absolutely. What advice would you give if you could time travel? And let's say you're going back to the 18-year-old Roberto. So this is before he's embarking on his college career. And I'm assuming 18. That's when you graduated high school. So let's say you're 18 years old, and you can go back to yourself. What would present day Dr. Malinow tell 18-year-old Roberto?
Enjoy the ride. Yeah, there's going to be lots of up and downs and lots of things that happen, and try to just take it all in and enjoy it.
Beautifully stated. Is there anything else, any last thoughts, comments, anything else important you want to share that I haven't asked?
No, I just really hope that your viewers will take it up and try to see if activating these negative thoughts and negative feelings in both depression and pain research that I think could test this idea.
All right, thank you. And if people are interested in learning more about you, do you have any websites or social media platforms anywhere where people can learn more about
Yeah,
you?
I have a website that has all my publications, and so that's pretty much a list of everything that I've done is on that website.
All right. And what is the website? Do you have it? Or we can also
Um,
put it into the show notes as well.
yeah, well, it'd be under UCSD somewhere. I'm sure that there's some website associated with UCSD that has all my publications.
Excellent. So we'll put a link in the show notes for Dr. Malinow's website. And I think with that, it's been a real honor interviewing you. I really appreciate you taking the time to communicate with me via email. I know I was just did a random email asking for the paper, and now it's coalesced into this interview. And I really appreciate all of the scientific inquiry that you've done. And like you said, that this is how science progresses: is one research, one hypothesis, replicating it. And when I see the work you've done, and not only the work you've done, but the others that you've mentored throughout your career, it's means a lot. And I really appreciate you taking your time to be here today.
Thanks for joining us for this fascinating deep dive into the basic science aspect of ketamine therapy with Dr. Roberto Malinow. His discussion regarding ketamine targeting hyperactive circuits, but only when those circuits are activated, presents intriguing research questions that could inform future treatment development. For providers and researchers, this work highlights important questions about whether our current approaches are optimal and suggests specific research directions worth pursuing. If you found this episode thought-provoking, we really appreciate it if you would leave us a review on Apple Podcasts or Spotify. It helps other medical professionals discover these important conversations. For complete show notes, including links to Dr. Malinow's research, visit ketaminestartup.com/podcast. And remember, the information shared in this episode is for educational purposes only and does not constitute medical or legal advice. Treatment approaches discussed should be evaluated with your specific practice context and regulatory environment. Thanks again for listening to the Ketamine Startup Podcast, and we'll see you next time.